Hi, my name is Ed Stowe, and I'm a graduate student at UGA. And today I'll be talking about the analysis my co-authors and I have done, looking at whether it's possible to use publicly available data to assess the effects of stream restoration on fish communities. So stream restoration encompasses many activities, and this includes the placement of in-stream structures, remeandering of straightened channels, armoring of banks, replanting riparian areas, and these activities are being carried out at a large scale with tens of thousands of projects and billions of dollars in spending annually. And importantly, a lot of stream restoration is carried out to offset losses to other streams, as required by Section 404 of the Clean Water Act, in which unavoidable impacts to aquatic resources must be mitigated. But the underlying assumption of compensatory mitigation is that if we damage a stream in one location, we are able to actually bring about benefits to stream ecosystems in other locations. And but it begs the question: does restoration actually improve stream ecosystems in areas, for example, like water quality, in-stream habitat, sediment retention, etc.? So far, there's really mixed evidence for this. And one barrier that's been cited to our understanding about whether restoration improves stream ecosystems is a lack of monitoring. So, for example, one study um, suggested that only about 10% of projects monitored habitat, biology, or ecology. However, in the case of mitigation banks, monitoring of biological and other parameters is compulsory and is documented and submitted to regulators in the form of banking instruments before restoration is done, and then post restoration monitoring reports in the years following restoration. And furthermore, these documents, as well as other data related to restoration projects, are housed in a database that the Army Corps developed called the Regulatory In-lieu Fee and Bank Information Tracking System, RIBITS. And so what RIBITS does is it potentially creates an opportunity to explore whether stream restoration projects are effective at a large scale. So the broad question that we're interested in answering with this talk is it possible to assess the efficacy of stream restoration using publicly available monitoring data on RIBITS? Now, a lot of different parameters of interest, both biological, hydrological, etc., are collected during restoration monitoring, but we zeroed in on one that we wanted to focus on, which was fish communities. So our specific objective was to determine if RIBITS data permits an assessment of how effective stream restoration is at improving fish communities in Georgia. We focused on fish for a couple reasons. First, improving the fish communities is a frequent goal of restoration. And when we say improving fish communities, we really mean increasing the diversity and abundance of fishes, and in particular, threatened or imperiled sensitive taxa. Fish are frequently used as a bioindicator, which means that they're frequently used to assess whether an ecosystem is improving or is degrading. Um, and finally, fish conservation is generally of more interest to federal and state authorities who are tasked with managing threatened species. And so it would be great if we knew whether stream restoration is actually useful for these taxa that we do manage. Fish monitoring at Georgia mitigation banks occurs in most projects or has occurred in most projects before 2018. Some exceptions are cases where watershed areas are less than 1 square mile. So these are very small streams. The monitoring is typically done pre-restoration and then in odd years following restoration. So years 1, 3, 5, and 7. And IBI scores have generally been required. These are indices of biotic integrity. So it's kind of an aggregate score that represents the fish community. Starting in 2018 and onward, no fish monitoring is now required under the Savannah District's new SOP for compensatory mitigation. So the methods that we used to do this study were first we wanted to evaluate how much monitoring has occurred. So we did that by collaborating with our colleagues at the U.S. Fish and Wildlife Service as well as consultants to compile as much information as possible on mitigation projects to identify projects with fish monitoring and in-stream restoration work, and determine, from these data, the extent of fish monitoring at each project. So basically collect a bunch of data that's not publicly available um, to understand how much fish monitoring has actually occurred. And then once we've determined when and where fish monitoring has occurred, we wanted to go onto RIBITS and actually assess whether the data for those fish monitor events was available on RIBITS. Furthermore, since we're ultimately interested in whether fish communities change following restoration, we also extracted fish sampling data from a subset of projects to determine if the availability um, of important sampling metadata like length of sampling and date of sampling, which these are the kind of data that might enable comparisons of fish communities before and after restoration. So I'm going to go into the results now, which are basically broken into three sections. I'm first going to talk about the sites that we used in our study, then how much fish monitoring actually occurred at these sites, and then finally, how much monitoring data was available on RIBITS based on how much fish monitoring occurred. So we identified 35 stream mitigation bank projects out of the 118 on RIBITS to really zero in on. These 35 projects included 111 different reaches with fish monitoring. So the projects ranged from having one study reach to 17 at one of the larger sites, with the median being 2 per project. And we tallied about 305 fish monitoring events at these 35 stream mitigation projects. These 35 projects were restored from approximately 2004 to 2017, with the bulk of the projects occurring from 2009 or so to 2014. So I'm going to talk about how much baseline fish monitoring actually occurred. But before I go into details, there are several figures like this. So I just want to point out that on the x-axis here, this indicates the restoration year, with the baseline year being before restoration occurred, and then year one being 1 year following restoration up through year 7, which is typically the last year that monitoring is done. And you can see here that fish monitoring was very common before restoration. So it occurred at 84% of the sites in this baseline time period. Fish monitoring was less common in years following restoration. So in any given year following restoration, we were able to document monitoring in about 50 to 70% of cases. As you can see, we had some difficulty determining if monitoring had occurred, particularly in years 5 and 7, represented by the green area. So this is how much monitoring we think actually occurred, but we're interested in how much of this data is available. So I'm going to zoom in now on just those cases where fish monitoring occurred. That's where we wanted to go onto RIBITS and see how much of this data was available. So as you can see, pre-restoration data was widely available. About 93% of the sampling events that occurred before restoration had some fish data, and 55% of this was raw data. So raw data is counts and abundances of specific species of fish, and this is most useful to researchers in terms of documenting how communities are changing in response to restoration. The bulk of the remaining monitoring data was largely in the form of quantitative fish metrics. That's the green areas on the plot. And quantitative fish metrics encompass things like the number of different species, the identity of the different species, or these IBI scores, as I've mentioned, these are still possibly useful, but not to the same degree as raw data. So following restoration, the post-restoration fish monitoring data are considerably more scarce. We see that monitoring reports were lacking um, for over 50% of sites in all years. And this amounts to fish data being available for only about 39% of post-restoration sampling events. And furthermore, only 22% of these post-restoration sampling events contained raw data. So in order to study how effective restoration is on fish communities, we generally need data from before and after restoration. So are these comparisons actually available using the data that we found on RIBITS? Well, we look at this in terms of both any type of monitoring data before and after and raw data. So we see that 30 sites out of our 111 sites, that's 27%, had both pre and post data. Um, this includes 20 sites with post data from years 5 and 7. And I'm emphasizing years 5 and 7 because these are probably of more interest because it's thought that fish communities might take several years to reach a more of an equilibrium community. So these are the monitoring years of greatest interest. When we look at how many sites have raw fish data both before and after restoration, it's only about 9%. Um, and this includes only 6 sites with post data from years 5 and 7. So it's really only a very small percentage of the sites that we've looked at are we able to compare the fish communities before and after. Lastly, we looked for metadata that would help us compare sampling events from before and after restoration. And we see that length of sampling information is missing for 44% of surveys, and season of sampling is missing for 9% of surveys. And so this missing metadata further hampers our ability to determine the effectiveness of stream restoration. So a core concept in the ecological restoration literature is the importance of the quantitative evaluation of restoration objectives. And when we looked at whether we could make these types of quantitative evaluations on improvements to the fish communities using RIBITS data, we see that that's not really possible. Um, Furthermore, it's likely that these results are applicable to other biological or hydrological parameters of interest because those data are also typically found in monitoring reports, which were not available to the extent required to make these types of comparisons. And finally, we also saw that lack of other ancillary metadata like date of sampling, um, and that the lack of those data also make comparisons difficult. So just to sum up, we know that these data are being collected and have been collected at massive scales, and this requires substantial investments of time, effort, and money. But in order to harness all that data that we're collecting, it's clear that some additional effort is needed to make sure that data collection is as standardized as possible and that the data is disseminated as widely as possible. And doing that should enable us to assess whether stream restoration is effective and whether stream mitigation banking is truly offsetting losses to streams. So with that, I'd like to thank our funding source, the USGS, um, who funded this project as part of the Science Support Partnership Program, which is a collaboration that directs USGS funds to USGS scientists for research and technical assistance projects that US Fish and Wildlife Service employees propose. And in this case, we need to thank Robin Goodloe, who was the person from US Fish and Wildlife Service who proposed us looking at fish and stream restoration. And we'd also like to thank her colleagues at the Georgia Ecological Services Office, Martha Zapata and Scott Glassmeyer, who have been instrumental, and also all of these other folks that you see here. Thank you very much.